So today topic is cheminformatics. It is very important what you have already learnt previously, how you are arrived that particular thing, and how you have gained the advantage in the CAD. So we were able to know about the advances, how the computer-aided drug design has came into the existence because of the cheminformatics. So what is cheminformatics? Cheminformatics, chemical informatics, it is also called as a computational chemistry. This is where we are using computer to make the things better with respect to chemistry. So it is a set of the computer algorithms and the tools to store and analyze the chemical data. So we are going to store the chemical data and also analyze the data in context of the drug discovery and designing the projects. Drug discovery and the design. We want to make invention of a new drug and discovery. This is what we have already used previously, and we have written some questions also, and you have done the quiz also. So why cheminformatics is important to drug discovery? Anybody can answer this question? Yes, somebody has already answered. Four already answered it. So there are four options are there. Why cheminformatics is important in the drug discovery and design projects? You have to answer this. Abhinay answered, Anish answered, Bhumi, Akshaya answered. Javali, Shreya did not answer. This is the just it is the definition part of it we are learning. Rohit has already answered. Jahnavi, Meghana answered. Chandan, Vaishnavi. Okay. The one which you need to know is it helps to store and analyze the chemical data efficiently. That is the reason we are getting the advantage. It helps to store the analyze and chemical data. So some of the questions what you are having is you need to write some matter, so just like a fill in the blanks. You have to write also. So the correct answer is it helps to store and analyze the chemical data efficiently. Because somebody is not attempted it, I'm canceling now, ending the question. Immediately try to attempt it because our class will be delayed. Only 15 have answered. The correct answer is it helps to store and analyze the chemical data. So next question, be ready to have it. Otherwise, it will end immediately when you don't respond. Ask remaining people also start joining the class. So What else the chemoinformatics informs? That it is the mixing of information resources to transform the data into information. So I think now you might have already got the data and the information. So don't get confused. We are getting a lot of data that will become an information if we process it. So that will convert it into a knowledge. So we have got a plenty of the data, like what are the temperatures which are present in this universe and the world. So this is the data. And you want to make a trip to somewhere. So you want to get an information that you are going to have some place. The data will be used to get that information. What is the temperature conditions? So then this will be used to put your clothing, and because this help you, this information become a knowledge to you to take a next action, like what type of clothing, what type of number of clothing, and what you need to take care because of the temperature. So this knowledge will be used. So the data is converting into information. That information creates the knowledge. Because of that knowledge, you take an action to pack your baggage. Similarly, it is a chemoinformatics is the mixing of information resources to transform the data. What we have got so many fragments, it is present in the form of some moiety or molecule, and convert that information into a knowledge where we are creating a drug. So for the intended purpose of making a better decisions, so that we can have a structure activity relationships faster in the arena of the drug lead identification and optimization, so that we will be converting the lead into a drug. This is what chemoinformatics helping us, because it has already stored the data. It is helping us to analyze that stored data so that we can process it. So chemoinformatics encompasses the design, creation, organization, management, retrieval, analysis, dissemination, visualization, and use of the chemical information. This is all the things which are, so we are creating the chemical information, whether it is a solubility or a melting point or molecular weight, or it is a electron density or substituent constant, log B values. These are all the things that encompasses. We design to get those, and we create those values and organize them in a proper manner and manage them to get used and retrieve whenever we require, analyze, so that it is correct or not with the substituents group what we have already there, and we disseminate to the students or the other research which are having the collaboration and visualization of the particular data, and use this information for the our drug discovery and design. This is actually what the informatics helping us. Now you tell me, which one of the following is not the function encompassed by the chemoinformatics? Answer this question. As 4 I have already answered out of 27 students on the board. Aditya T, you try to answer. You have to see not the function. Only 15 have answered. As Aditya, you have all answered. I think you have done in 2 devices. Kiran Teja already answered. Akshaya answered. Ashmita, try to answer. Meghna, Pranati, Sahit answered. Javali has answered. Sivaram Prasad, you have not yet answered. Srivalli and Shruti also. Because if you delay it, we will lose our remaining slides. We will have to move fast. Immediately try to answer. Be respond immediately. So I am moving to the next question. So I will end up the question now. The last few minutes are there. If somebody wants to answer, now only answer. Only 20 have answered out of 30. Somebody are inactive or kept the phone aside and they cannot answer. It will be visualized here. So I am ending the question now. So now you see what is the correct answer. So we don't use this data for the cooking food because our drugs are so complex. We use this particular chemistry data where our cheminformatics, everything will be known by our mother in the kitchen. She manages this cheminformatics without use of any computer and mobile. She cooks food to up to the mark. We don't use it for the cooking food. Remaining all we are going to use. That is the reason it is not the option. So we are moving to the next question. Next slide. So why we need cheminformatics? We have already said that even though we are getting in the kitchen so many chemical compounds and the food, there are all the chemicals which are also having a chemistry. We are able to manage it. But why we need a cheminformatics is that what is invisible, we are managing now. The chemical structure of a protein invisible to our naked eye, and the chemical molecule which we are acting as a ligand to treat the disease also invisible. Both are getting interacted, that is invisible and very difficult to count. That is the reason we need this cheminformatics. So cheminformatics is essential in the modern drug design because it transforms raw chemical data into actionable knowledge. Simply having a data of temperature of different places in the world, it will not solve the problem. And unless otherwise we categorize them, we take the linking of this data to the particular travel, and we're taking a decision, and it will not be converting into a knowledge and utility. Simply generation of the data is not good. Similarly, what we have created a chemical information that is not sufficient, we need to convert it to the actionable knowledge so that we will resulting into a better drug design, and we are going to yield a improvement in our efficacy and reduction in the toxicity. So it allows the researcher to navigate the vast chemical space of 10 to the power of 60 potential drug-like molecules. Imagine how many number. After 10, you are getting 60 zeros. How many number you are going to get? That many chemicals it is processing. That cheminformatics. So then the bench chemistry, what we are doing in the lab, what we are getting, it is very small number we can execute in our lab. But it will do 10 to the power of 60 potential drug-like molecules, and it will be giving as a knowledge to us. It is cornerstone for the reducing the high cost and lengthy timelines. So we reduce the cost also, lengthy timeline also in bringing a new drug to the market. So it is being used in our CAD. Till now, what we have got 3 units. They are all the 3 based on this particular 4.1 unit, that is cheminformatics. So by performing many of the initial screening and optimization step virtually or in silico, it is possible to do without entering into the lab. Now you have to type the answer. Fill in the blank. The cornerstone for reducing the high cost and lengthy timelines of bringing a new drug into the market is, you have to type the name. Just you type the name. It is a fill in the blank. 4 have already typed. Abhinay, you type. A single word is sufficient to write. This is the mere topic of today. Ultimately, what we are learning. Five have already answered. Akshaya answered. Sahit. Shreya answered. Tejashvi answered. Chandan answered. So 10 have already answered. Now I will see what is the correct answer. So the cheminformatics is the correct answer. This is actually what is reducing. It is only the reinforcing your learning. Somebody has written computer-aided drug design, but it is a broad topic. In that, cheminformatics is the one which is making. So many had written CAD also. Somebody has written cheminformatics like. So cheminformatics is the correct answer. Yeah, very well. So virtual screening and hit identification, the research used the computational models like Autodoc, Vina, and Glide to assess the millions of the compound virtually. Millions of the 10 to the power of 60, it is already available virtually without going to the lab in the computer in silico, identifying those most likely to bind the biological target before their physical experiment is conducted. That is the reason this cheminformatics helping virtual screening and hit identification. Predicting the ADMET properties. So it identifies the compound safety and effectiveness. Then only it will be druggable, drug likeliness is more. Early by predicting the absorption, distribution, metabolism, excretion, and toxicity. Tools like ADMET SAR, ADMET Lab. So we are using based on this particular data also filtered out the candidates likely to fail the clinical trials due to the toxicity or poor metabolism. So we are finding failures fast. So in this quiz also you have seen fastest finger first. We are entering into the market first. They are going to winners and they earn money more fast. That is the reason we need to be little bit alert also when we are having it so that we can compete with others. Structure activity relationship modeling can also be done by this particular cheminformatics by analyzing molecular fingerprints. Just like a fingerprint, each individual is having specifically, each molecular has got some properties that act as a fingerprints to them. So melting point, solubility, its color, and its substituent groups, log P values. So what you have got steric factors, these are all the fingerprints of the particular moiety. So QSAR models, chemists can predict how specific structural changes like adding functional group will affect the drug biological activity. So we can have the QSAR also by this cheminformatics. So data management and mining, it enables the storage, indexing, and rapid searching of the erroneous, enormous databases, which we don't use this one, it will become erroneous later. So what that such a database is? PubChem, ChemEmbl, EMBL, expanded form. So our drug bank, which is making decades of the chemical research instantly accessible and making our time saving. Machine learning and algorithm, AI, artificial intelligence integration, advanced algorithm, including the deep learning and random forests, where we will be picking up the desired moiety, where depending upon the need. So it is having well versed with all. So we need to pick up the best suits to us. Now used to find the complex pattern in the big data that human researchers might miss. So we find a rare drug which is already present in the database. We can pick up from that particular random forest what is utilized for our requirement, further accelerating the lead optimization and the process. So now you answer this question. Which of the following are applications of the cheminformatics in the drug discovery? It is very easy question. If somebody has attempted, so multiple answers are allowed. Now you can answer all possible one. Then only you will get a correct. The negative one you have to leave. Remaining all you have to write. So this will become a 2 marks question to your exam. Applications of cheminformatics in drug discovery, drug design and drug discovery. Eleven have already answered. So if 10 members are answering means I will go to the next slide. So what the screen which is showing is the correct answer. So this is used to the for the visual screening and hit identification, where we are having a lead or chromophore identification, predicting ADMET and structure activity relationship modeling. For this purpose, we are using this. So I'm going to the next slide. 17 members responded out of 31. What are the essential cheminformatic tools? Research use a wide variety of a toolkit to manipulate chemical data. The most significant is RDKit, 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 a versatile open source library that serves as a Swiss Army knife for the field. So this RDKit is used for the general cheminformatics, molecular drawing, descriptors, calculation of log p values, and molecular weight calculation based on the atoms which we have already given in the structure, fingerprint generation for the machine learning by generating a smile and collecting the data, assigning the data to that particular smile file. So OpenBasel is another one which is used in the cheminformatics for the file conversion. It converts between 110 plus chemical file formats, smiles to SDF, and what we are having a human readable form to a computer readable format. Later onwards, it will be converting into a bits. So it will be using in the file conversion. DeepChem is for the deep learning. It provides the pre-built model for property prediction and molecular generation. So this is actually a little bit advanced. We can have a property predicted without going to the lab. How we have done the substituent analysis, and we are able to predict the log p values in our OBT you have done because of this particular deep learning. What it is been done, what you have done in the lab bench top on the class, it will be done by the computer, give the value to you directly. Data Warrior, it is a data visualization software, combines the chemical intelligence with the interface data plotting to analyze the large compound libraries. So it is an interactive data plotting. So here it will give the relationship between one factor to another. CDK, that is Java Toolkit, a foundational Java-based library used for the structure representation and SIR analysis, that is quantitative structure activity relationships and your structure activity relationships. Now, which tool is primarily used for the file conversion? Just if you want to go, I will go back to the previous slide. Just you read it. For your information, I am showing you. You read it. Again, reinforce your learning. So now you answer the question. What is the right answer in the question? Aditya T answered, Abhinay answered. So 10 persons have already crossed. We will be moving to the next slide. Now we will see the responses and I will tell you on the screen what is the correct answer, which is appearing the screen is OpenBabel. It will convert file if you want to have conversion of your chemical structure, that is IUPAC name to or your ring structure to Smiles data. The software can be used in the Cheminformatics OpenBabel. So write it fast. I will be moving to the next slide. So why we use this type of toolkits and this Cheminformatics to handle the large amount of information? It is beyond our human brain to remember. To move the chemistry to the computer age so that we can convert this into the machine learning and artificial intelligence. To move the data to a knowledge. Data is lying down without any usage. Knowledge is only having an application. That is the reason we are having these tools and this field. So how this computer is learning and what is the learning pace what we have got now. So there are two types of the learning which will be present either by our students or teacher or by the computer. There is a inductive learning and deductive learning. So one is proactive learning and it is a passive learning. Active learning and inactive learning. So what is that? We will little bit seeing it. So the first one is the deductive learning. It is a fundamental theory exists which allows us to calculate the properties and predict the behavior of the molecules. So the fundamental theory for the chemistry is a quantum mechanics. So this is the fundamental theory based on the facts, learning it. And we are proceeding further, which is already existing. Based on that, we are proceeding. That is called as a deductive learning. So now this answer, this question, because it will become next 5th unit, this is you are going to learn. Type your answer. If you want to go back and see in the one, I will just again go back and show you. And read it carefully. So it gives the importance that you need to be alert throughout the class. Even you are missing it, so you will lose some knowledge. So just getting a data by mugging up is not sufficient. You need to apply it. So now you answer this question. Levin has already got. I will show you what the answers. First, view the correct answer. So this is quantum mechanics. Based on that, we have already drawn all these toolkits and Cheminformatics in the computer. This is quantum mechanics. And you see the correct answer and show the answers. What somebody has written. Everybody has written quantum correct exactly. You are smart. So the quantum mechanics, the deductive learning used this quantum mechanics, that is fundamentals, and it is deducted some learning. So basis for it is what is the fundamental to that chemistry what we have got. The basis for that fundamental is a quantum, where we have got a deductive learning. This is what we need to understand. So inductive learning is active learning. So you create something. Whereas deductive learning is learning from the examples, which is already present in this world. So this is what the fundamentals which is already been created. Based on that, we are creating, that is deductive learning, but you are creating new concept and you are making above it. That is de novo, that is inductive, which is not existing previously. So where you are making it new, that is inductive learning, active learning. Whereas learning from the example, so which is already been there, you are mimicking the same thing. That is actually a deductive learning. Say, for example, now I'm going back and showing you what the slide previously you see and answer. This is actually what the deductive learning is. You are learning by example and seeing it, and you are doing. Because that based on the data only you are able to answer. Something which I am asking like a proactive and imaginatory creative questions. There is no answer for it, but you are writing. Such things is called as a inductive learning. Now, till now, we have not done that particular inductive learning where you are creating something and giving a teacher. Now, what you are doing is you are learning from teacher and making it with a example. So this is basically differences. In our computer also, we have got supervised learning and unsupervised learning. Supervised learning is a deductive learning. We give the example to the computer. It will understand what is the pattern and it will give the correct answer to it. In inductive learning, so the computer learn actively and predict, that is projects or predict something and which is beyond our human scope, it will give it. That is actually unsupervised. It is beyond our, we cannot imagine such a unsupervised learning created wonders. So in the form of hidden layers, it will create and give the output to us. So both the learning are very important. We need to understand the concepts. In the 5th unit, we will go further. Don't worry. So here is the object we are giving as input, and this deductive and inductive learnings are present as a hidden layers in our statistical patterns or methods or neural networks, which are hidden layers, and it will give as output to us. It is a property in our cheminformatics. So that is either a biological activity prediction based on what we have got a structure. So ultimately, what we are getting an object, we are inputting it, getting an output, and learning method, either it is a inductive or it is a deductive. Which of the following are the general scheme for the inductive learning? Outputs. You have got a structure, you have got a learning method, you have got an object. So what is you have got output? We see the correct answer. Based on the property, what you are creating with that structure, you are creating a property. You are giving a structure, substituent groups are taken and it gives the log p value as a property. It is an inductive learning. It is already not, we don't have log p value, which we have not done for a new compound, which is we have not seen and which is not present in the library. We have fed to the computer as a structure or as an input. Then it is giving a property of its solubility, melting point, log p values. This is property. It is actually what called as a inductive learning, which is not existing previously. There is another one. Is we keep the data for the carboxylic acid with homologous series as a benzoic acid and substituents. We are giving all the data and asking it, and it matches what the correct answer it will give. That is called as deductive learning. So you have to compare and know what is the difference so that you will have a machine learning methodologies also. Next. So the fundamental tasks of a chemist Based on that, only we have already proceeded. The chemist will have the property predictions. Later onwards, he need to use the prediction for synthesis feasibility, and he has to design the route or the structure better, and reaction predictions, rate constant predictions, and structure elucidations. So these are all the things which the chemist required to done. Based on the chemical structure, he will have a physical property, whether it is a liquid or solid or semi-solid or color or flavor, how it is appearing obnoxious or pleasant, and he can know the chemical, he can have the biological activity, and quantitative structure activity relationship, quantitative structure property relationship, QSPR and QSAR, both which are linked to your biological activity and structure elucidation, reaction predictions. So by giving the available material, starting material, it will give the synthetic design, how to get a, how many paths are there to get a product. So these are all the chemist. So they bound to use for all these cheminformatics without any file. So the realm of cheminformatics, understand and be careful. Representing the chemical compounds, searching the chemical structures, similarity searches, relating structure to the properties with the models. What is cheminformatics doing? It is representing chemical compounds. One is a database. So, and these chemical compounds with the structures, it is giving and converting into a different file formats and everything. Similarly, it is having a similarity searches. What are the compounds which are having a similar in with respect to some lead or a basic moieties or chemophore or this pharmacophore, and relating to the structure properties with the models, relating the structure to the properties. So this is what the cheminformatics doing. I think you might have already seen, understood this. We are going to the next slide. Which one of the following are included in the realm of cheminformatics? Multiple answers are there. You can answer 2 or more. I now already answered. I'm going back. See, representing the chemical compounds, chemical structures, and similarity searches, providing the properties with the models. So you the correct answer. Representing the chemical compounds and similarity, searching the chemical structures and similarity searches. Go back and reinforce your learning. So it is concerned with something chemical compounds and its file formats, similarity of the chemical compounds, and relating to your properties. Nothing to do with your reaction rates. Later onwards, based on this data, you will have that reactions. Kinetics, everything will come a little bit later. These predictions based on the inductive learning of this, you will be able to get later. So the answers. So I will move to next because 16 I've already answered. Machine learning methods. Important role in the cheminformatics to yield go further is, for example, it is usually difficult to predict which type of the descriptors are most suitable for a given search or the classification we want to get output of a particular property. We want to have a biological response. So therefore, the machine learning techniques are often used to facilitate the descriptor selection. So machine learning, it is not how it is making learn. It is taking the data what we have given as a structure, and it is converting into a smiles what we have provided. It is taking as a bits, and it is taking that one into a individual descriptors, and the descriptors are stored later onwards when we use for the further. This machine is already learned with that particular compound having structure, which is having say so many descriptors like a log p values and your partition coefficients. So these substituent constituents impact, SAR, what you have got linkages with the steric factors, electronic factors. These are all descriptors which are present. So machine learning methods, they are belonging to genetic algorithms. So different parameters and model solutions to given problems are encoded in the chromosomes. We know in the genetics, and based on the chromosome only, we have got a personality traits and the color and complexion and whatever the which is properties which is giving an output based on the input what is present there in the ovum, and the spermatozoa which is resulting in a zygote that is ultimately present as a chromosome. That is making the single cell converted into a genetics, and making the genetic one present in all the body, all cells are the same. So this similar manner, this algorithm uses particular descriptors what we have given and uses further. That is the reason it will not miss and it will not commit any error if it is doing perfectly. And subject to the random variation that's generating the population differences, it will learn that mutations also, changes also, how it is impacting its property. And solutions are provided by these chromosomes are evaluated by the fitness function that assign the high scores to the desired result. Similarly, the cheminformatics also changes the functional groups and similarity searches it will make and compare it, and it will assign the scores. Chromosomes yield best intermediate solutions are subject to the mutation and crossover operation that corresponding to the random genetic mutation and the gene combination events. So you will get a multiple diverse data based on these changes, and the resulting modified chromosome represent the next generation process. So what we have got a modification and we have got a descriptor changed with respect to the modification, which will be used for the new drug discovery in the de novo drug design. Continued until obtained results are satisfactory convergence to yield a perfect drug moiety and perfect human being, or the chromosome which is representing these mutations which will be coming out as an output. Similarly, which we are having a changes in our chemical property, which is resulting into a definite output that is having a biological activity. This is what the machine is doing, learned for our purpose. So now we will be, before entering into the topic, there is a small announcement. We will have remaining thing in the next class. 20 more slides are there. And tomorrow we will have continuity and ask your remaining people. At 2:10, we will have the remedial class. There we are going to discuss, anybody can attend, and those who have got less than 8 marks should certainly attend remedial classes at 2:10. Another one is we are going to have a 5 minutes mentor-mentee meeting that is mandatory to know what we are learning, what modification we have to do it. So at 2:45 we will be meeting, 2:40 we will be completion of this remedial class. We will be meeting and finishing within 5 to 10 minutes. That is by 2:50 we have to complete mentor-mentee meeting. You tell your remaining mentors also 1 to roll number 13. They can join the mentor-mentee meeting at 2:40. So we will have a meeting and wind up today's session. Tomorrow we'll meet again with respect to your continuity of what the Cheminformatics helping in the QSCR. Now I will open the floor for the discussion. Anybody is having a doubt, they can unmute yourself and ask the questions.